Hello guys, welcome to my channel Learning Biophysics with Team 320. The first topic for today is ionic channels. Ionic channels are transmembrane proteins that form pores within cell membranes, acting as critical regulators of ion flow. These microscopic gateways play an important, indispensable role in a vast array of physiological processes, including nerve impulses, transmission, muscle contraction, and hormone secretion. Their malfunction can lead to a variety of diseases, underscoring their significance in maintaining human health. Structure and function. Ionic channels are typically composed of multiple subunits that assemble to create a central pore. This pore is selective, allowing only specific types of ions to pass through, such as sodium, potassium, calcium, or chloride. This selectivity is determined by specific amino acid residues lining the pore and channel's overall structure. The opening and closing of these channels, known as gating, is tightly regulated. Various stimuli can trigger channel gating, including voltage channels. Voltage gated channels respond to alterations in membrane potential, a fundamental mechanism in nerve impulse propagation. Ligand binding. Ligand gated channels open or close in response to binding of specific molecules such as neurotransmitters. Mechanical stimuli. Mechanosensitive channels react to physical forces such as pressure or stretch, playing a role in touch and pain sensation. Types of ionic channels. There are numerous types of ionic channels, each with unique properties and functions. Some prominent examples include voltage-gated sodium channels, essential for generating action potential, the rapid electrical signals that travel along nerves. Voltage-gated potassium channels contribute to the repolarization phase of action potential, restoring the resting membrane potential. Calcium channels play a vital role in muscle contraction, neurotransmitter release, and gene expression. Chloride channels involved in regulating cell volume, pH balance, and transport of other ions. Physiological significance. The proper functioning of ionic channels is paramount for various physiological processes. Nerve impulse transmission. The coordinated opening and closing of voltage-gated sodium and potassium channels generate action potentials, enabling rapid communication with the nervous system. Muscle contraction. Calcium channels mediate the influx of calcium ions into muscle cells, triggering the interaction between muscle proteins and leading to contraction. Hormone secretion. Calcium influx through voltage-gated calcium channels triggers the release of hormones from endocrine cells. Sensory perception. Various ion channels contribute to our senses, including touch, pain, vision, and hearing. Clinical relevance. Dysfunction of ionic channels can have profound clinical consequences. Genetic mutations or acquired abnormalities in channel function can lead to a wide range of diseases known as channelopathies. These include epilepsy. Mutation in sodium or potassium channels can disrupt neuronal excitability, leading to seizures. Cystic fibrosis. A defective chloride channel leads to the buildup of thick mucus in the lungs and other organs. Cardiac arrhythmias. Abnormal heart rhythms can result from mutations in cardiac ion channels. Muscle disorders. Myotonia and periodic paralysis are examples of muscle diseases caused by ionic dysfunction. Research and future directions. Ionic research remains an active area of research. Scientists are continually striving to understand their intricate mechanisms, develop novel drugs to target channel dysfunction, and explore their potential as therapeutic targets for various diseases. The study of ionic channels holds immense promise for advancing our understanding of human physiology and developing innovative treatments for a wide range of medical conditions. Now, my second topic is synaptic transmission. Synaptic transmission is the fundamental process by which neurons, the specialized cells of the nervous system, communicate with each other. This intricate process allows for the transmission of information throughout the brain and body, enabling everything from thought and perception to movement and bodily functions. The synapse. The synapse is a specialized junction where communication between neurons occurs. It consists of three main components: presynaptic terminal, the end of the axon of the sending neuron; synaptic cleft, the small gap between presynaptic and postsynaptic neurons; postsynaptic membrane, the membrane of the receiving neurons. Steps involved in synaptic transmission. Synaptic transmission involves a series of carefully orchestrated steps. Action potential arrival. An electrical signal called an action potential travels down axons of the presynaptic neuron, reaching the presynaptic terminal. Neurotransmitter release. The arrival of the action potential triggers the opening of voltage-gated calcium channels at the presynaptic terminal. Calcium ion influx causes synaptic vesicles containing neurotransmitters to fuse with presynaptic membrane. This releases neurotransmitter into the synaptic cleft. Neurotransmitter diffusion. The released neurotransmitter diffuse across the synaptic cleft and bind to specific receptors on the postsynaptic membrane. Postsynaptic potential. The binding of neurotransmitters to receptors causes ion channels to open or close on the postsynaptic membrane. This leads to change in membrane potential of the postsynaptic neuron, creating a postsynaptic potential. This can be either excitatory or inhibitory. Signal transmission. Signal termination. The neurotransmitter signal is terminated by either reuptake back into presynaptic neuron, enzymatic degradation in the synaptic cleft, or diffusion away from the synapse. Types of neurotransmitters. There are various types of neurotransmitters, each with specific effects on postsynaptic neuron. Some common examples include glutamate, the major excitatory neurotransmitter in the brain involved in learning and memory; GABA, the major inhibitory neurotransmitter in the brain involved in reducing neuronal excitability; acetylcholine, involved in muscle contraction, learning, and memory; dopamine, plays a role in reward, motivation, and movement; serotonin, involved in mood, sleep, and appetite regulation; synaptic plasticity. Synapses are not static structures, they can change their strength and efficiency over time, a phenomenon known as synaptic plasticity. This plasticity is crucial for learning and memory formation. Two important forms of synaptic plasticity are long-term potentiation, a long-lasting increase in synaptic length, thought to underlie learning and memory; long-term depression, a long-lasting decrease in synaptic strength, important for refining neuronal circuits. Clinical significance. Disruptions in synaptic transmission can contribute to various neurological and psychiatric disorders, including Alzheimer's disease, characterized by a loss of synapses and impaired neurotransmitters; Parkinson's disease, involved by degeneration of dopamine-producing neurons; depression, associated with imbalances in neurotransmitters like serotonin and dopamine; schizophrenia, linked to dysregulation of dopamine neurotransmission. Research in future directions. Synaptic transmission remains a vibrant area of research. Scientists are actively investigating the molecular mechanisms underlying synaptic plasticity, the role of different neurotransmitters in various brain functions, and development of new therapeutic strategies to target synaptic dysfunction in neurological and psychiatric disorders. Understanding the intricacies of synaptic transmission is crucial for unlocking the mysteries of the brain and developing effective treatment for brain-related disorders. Thank you.